My topic is on the newer technologies and the identification of the pathogenic organism. Now, why do we need the new technologies? Well, accurate identification of the pathogen underlying the infections is very, very essential. The other thing is a true turnaround time. That is very critical for treatment of any type of infections because it can progress to sepsis. And we know that every hour infection that remains untreated with effective antibiotics decreases the survival rate by 7.6%. Early identification of organism, early antibiotics, and changing the outcome. I think that is the main basis for the need for new technologies. Now, going back to the conventional methods, what we are doing or we were doing, we are culturing, reculturing as a pure isolates and then analyzing for the antibiotic resistance. That's what we have been doing. But the problem is the turnaround time is like 72 hours, 48 to 72 hours, disease can spread and can have complications. So cultures, they have a low specificity, sensitivity to identify the organisms that are nutritionally demanding, where biochemical activity is limited. For the organisms where you don't know the growth requirements or you need to know the growth requirements, the slow growing organisms, and where your blood sample is very inadequate, that also affects your results. The prior antibiotic exposure, that will affect the results, and contamination of the sample. Now, what is syndromic testing? Well, syndrome is a set of signs and symptoms, and testing means that you will group a probable pathogens which is causing a disease with one rapid test that will maximize the chance of getting the right answer in a clinically relevant time frame. Now, we have a lot of diagnostic tools, but they can be time consuming, labor intensive, technically complex, that may lack the specificity and sensitivity as well. Now, this is the traditional testing which we're using, and now we're going towards the syndromic testing. So one sample, multiple tests, multiple results, and then interpreting. So longer length of the stay of the patient. And syndromic testing, you take a sample, you get multiple results, and one report improves the patient management. Now, the newer technologies which I'll be discussing is a protein-based technology, nucleic acid platform and technology, sequencing of the ribosomal RNA, and next-generation sequencing. There are more biosensors and all, which I think was already mentioned. I won't be touching that. That's a latest one. The protein-based identification is antigen-based and malleative, which is the most important part of my presentation. Now, antigen-based identification is very simple to understand. Antigen, antibody, they combine together and you get a result. Basically, that's the main principle on which it works. So now this lateral flow assays have been used for a long time where the antibodies, they bind with the matrix through which the patient sample containing antigen, it passes, okay? You get a color, that is a calorimetric readout, which we have been doing for malaria, C. diff, and for HIV as well. So this is a basic principle. I wanted you to understand that what happens in this. You add a sample, it goes through the like conjugate release pad, which contains the antibiotics that are specific to the target analyte are conjugated to the color of fluorescent particles. You get a particular color. Basically, that's a basic concept on which your antigen antibody reacts. That's known as a lateral flow assay. Now, the new thing which has come, this colored result, which are your reading on a card, that has been replaced by a fluorescent labeled assays. That is much more sensitive than the lateral flow assays, basically. It's an automatic detection. Fluorescently labeled antibodies are detected. It has been used for bacteria as well as for the viruses. The other thing which is chemilucence. So that's the same thing. The fluorescent-based assay is also called a chemilucence, where antigen-antibody complexes are formed. There's a luminescent substrate which is added with an oxidant and enzyme. Basically, the luminescence which is produced, their intensity is detected. This has been used for hepatitis B, C, HIV. In our lab also, we're using the same test for our viral detection. That's a B, C, and HIV. So it's a very extremely sensitive test, economical way to quantitatively measure the antigens in different samples, and mainly for the viruses which we are doing it. Now, this has a new thing which has come as a 5th generation ELISA, which is again based on the antigen detection. We had a 4th generation and 5th both. The advantage with that is that you can detect a P24 antigen in the HIV virus. So Earlier, we used to depend on the antibody. We had to wait for the antibody to be generated and then, detect it. Now you can detect the antigen as well as the antibody. So early diagnosis in less than 2 weeks also you can get after the exposure. So that has revolutionized the HIV diagnosis and early management. Now, coming to the MALDI-TOF, which is very important and is being used now in almost every center, it's a matrix-assisted laser desorption, ionization, time of flight, mass spectroscopy. I think I'll explain it. So it is basically based on the assessment of a complement of ribosomal proteins as an isolate. So it identifies your bacteria at a species level. It can also identify the resistance enzymes. But yeah, we cannot detect the antibiotic sensitivity with it. It's very rapid, economical, and sensitive. This is the workflow of MALDI-TOF. I think we'll better understand from this. So we take a sample, cultivate it, take a colony from there, add a matrix. There's a matrix which can absorb the energy. So we expose that sample to the laser radiation. There is a generation of the ions and charged particles, right? So they travel through this tube. That is the time of flight. What do you mean by time of flight is that these particles, they travel through a tube. So depending on the mass and the charge, you will there's a detector who calculates the time of flight. So there's something you get like this, depending on the masses and the charges which are generated. And then you have a large database in which you can compare and identify the organs. That's a basic principle on which the MALDI-TOF works. That is coating the solution with the matrix, followed by laser exposure ionization, generating the accelerated protonated ions, and then analyzing it. So you basically detect the mass to charge ratio, and it generates a peptide mass fingerprinting, which you can compare with your database. Now, there are two things which we use, BRiCC MS and MALDI biotyper. It's the same thing, but they differ in the software. I just want to put a word about the BRiCC 2. So the BRiCC MS is based on the principle of the MALDI-TOF. Now we have BRiCC 2, which is not based on that principle. It's completely different. It's a fully automated system which can identify your bacteria as well as the antibiotic susceptibility testing, and it is based on the broth microdilution. It can give you a sensitivity report also. So this BRiCC system is based on the advanced calorimetry, basically the fluorescent-based method, where you will get a different colors when the different pathogens are exposed to the different reagents. That's a basic principle on which it works. It can identify bacteria, viruses, and you can also calculate the MIC of the organism. This has been used for a long time, the BRiCC. So this MS, which is based on the mass spectroscopy, using the MALDI-TOF, is a newer, faster. You can identify bacteria, fungus, yeast, and molds as well. It's cheap, very good sensitivity. So these are the two difference between the two systems. So now MALDI-TOF has been, is replacing it basically. But sensitivity still you depend on the BRiCC. Now the uses, I'll quickly go through all gram-positive, gram-negative organisms you can detect. You need only few colonies to identify the organism. You can detect it directly from the body fluids, respiratory sample, blood cultures, but usually in all the centers, we still depend on the cultures and then use a MALDI-TOF. From direct sample, it is very difficult because you have to isolate it, purify it. There is a long process to do that. It can identify the yeast as well as the fungus, which is not FDA approved, but still is being used. It can detect carbaphenemases, the MRSA, MSSA, VRE, beta-lactamases. Minimum processing time is within 30 minutes to 1 hour, you can get a result. Now, limitation is that you cannot use it for the viruses. The main reason is that the virus, they have a very high molecular, there's a very low protein content of the viruses. That's the reason we cannot use for that. For the fungal identification also, it's little difficult because of the strong cell walls. Coming to the nucleic acid based techniques, that is mainly based on the principle that each organism have a particular nucleotide sequence. So that is the thing which we should remember. Particular nucleotide sequence which you can identify, and that's the way you can identify the pathogenic organism. Now, the basic principle of nucleic acid probe-based assay is the intrinsic ability of a single-stranded DNA or RNA to anneal specifically to a complementary sequence and form a double-stranded DNA. That is a hybridization, where you introduce a sequence and that identifies a sequence of the pathogen and combines with that. So that's the basic principle on which the nucleic-based acid-based techniques, they work. So we have probe-based assays, multiplex assays, singleplex. I'll more concentrate on the multiplex assays. Quickly, probe-based assays is there. They are mainly based on the fluorescence again. You use the nucleic acids with the fluorescent markers. Better than the traditional DNA probes, rapid assay in less than 1 hour. This is a FISH technique which has been used. There are some developments in this. There's a quick FISH technique that has been combined with flow cytometry to give us the results quickly. So basically, this also is based on the same principle, hybridization between the specific fluorescent labeled probe and the complementary target DNA and RNA sequences. And you detect a fluorescent signal basically. So it can be DNA, RNA target or peptide nucleic acid targeted. So this is what happens. You introduce your primers. Primers are the sequences which you know and you are introducing to the sample. It identifies the corresponding sequence on the pathogen, combines with it, and it has a fluorescent tag attached to it. And you can detect the pathogen. That's a basic principle on which it works. You can identify bacteria, fungus, and mycobacterium. You can identify facultative bacteria, atypical respiratory pathogens, a large variety of the organisms. But for viral infection, again, it's not a good technique. Now, coming to the PCR. So what is PCR basically? It is the technique for rapidly detecting producing millions and billions of copies of a specific segment of DNA. So you amplify the DNA of the pathogen so that it is easier for you to detect the pathogen. That's the basic in simple language which PCR means. Okay. So what do? We first denature. So whatever is the DNA, you apply the heat. I won't go into the deep, like the process, how it is done. Denaturation means you will separate the strands. Okay. Then you introduce the primer, which combines with the complementary sequence on that. That is called hybridization. And then you add an enzyme, which is the DNA polymerase, which will synthesize it and amplify it basically. Now in earlier time or now also, like when you amplify, it has to undergo a electrophoretic process to identify the organism, and that process used to take time, which has been replaced now by the real-time PCR, where you can get the while you're amplifying, you can identify the organism. It can be a fluorescent dye base, and it has made life very easy. You can get the results very quickly. So that is called the real-time PCR or quantitative PCR. So I think I have explained it. Amplification, same. It's the same basic technique which was used many years ago, but now you can detect the real time. You don't have to go through a multiple steps of electrophoresis. That is the advantage of the quantitative PCR. So this is a singleplex or limited assay. I think I'll skip this. I want to go to the multiplex fast. The other thing is the digital PCR. This I found interesting. It is not being used yet, but the digital PCR, what do you do? Suppose you have a sample which has a very low concentration of the pathogen. So you can divide that sample into different wells, like multiple compartments, and then amplify it at that individual compartment. So your possibility of identifying the organism becomes easier. So it is still being made. I don't think it's available yet, but this is something which will make our life easy if you are trying to isolate the organism from the blood. You most of the time, and it will make our like the results and all you will get quicker. So this is the other advancement. This is a high resolution melting analysis. This is another post PCR which you can apply, and you can see at what temperature your DNA is melting. So that's a basic principle in simple language. You amplify your DNA, apply a fluorescence, and then you analyze it by melting it at the temperature. So different types of DNA will melt at different temperatures, and it will give you a curve that is matched with the database. So this is the basic principle of high resolution melting analysis. So that's the level of fluorescence and temperature will give you melting curves. Going to the multiplex PCR, where you can, more than one target sequence can be amplified by including more than one pair of a primer in reaction. Basically, whatever principle I've explained, you will add more than one DNA sequences and it will identify more than one organism in one test. So that is the basic principle on which multiplex PCR works. It saves your time, you get a multiple results. So you can detect viruses, bacteria, parasites, and different type of samples you can use it. Nested PCR is something which has made it more sensitive. So what do you do in nested PCR? You amplify DNA twice. So first the PCR amplification, then again the second step PCR. It makes it more sensitive. So 2 times amplification is done. That is called a nested PCR. It can perform 50 to 60 cycles of PCR. So more you amplify, in a simple language, more you amplify, more is the chances of getting the organism. And they are very highly sensitive methods. Now, this is something film array or a biofire which have been used in the lab. I found it very interesting because now, all patients, we send the samples for the biofire. Turnaround time is just 1 hour. So it's a one system fully integrated. There are some lyophilized powder form reagents which are present in the pouch, which is loaded. So basically, you first put a water inside or hydrate it and make it liquid. Then you add a sample in the next chamber, whatever you want to analyze, they all go into a pouch. So your sample as well as whatever reagents are required for the test, they all go there. After that, it is put in this instrument which is called a film array. Now this part I found very important to understand that how the biofire happens. So first is that it moves into lysis chamber. So what are you supposed, you need DNA and RNA. So first step is that you extract the DNA and RNA, or that's called a lysis chamber. Next, it undergoes the purification by the magnetic beads. And after that, it undergoes the amplification. One step amplification is done here. Then there's a next amplification which is done in a final chamber. So there is a 2-step amplification which happens, making it more sensitive. And based on the fluorescence, you can identify the organisms. And beside this, then they apply a melt is added and you get a melting curves on basis of which you can identify the different organisms in one test. So you can have a respiratory panel, GI panel, meningitis panel. It can also detect the resistant genes. That is the advantage with this. This, I think I'll skip this. The limitation of the PCR, it is highly sensitive, but the prior sequence data is required to compare, detects only presence of absence of known gene or pathogens. Primers can sometimes non-specifically attach to the sequences that are similar, but they're not identical complete to the target DNA. DNA polymerase used in PCR is prone to errors. Specificity is lower than culturing and staining. The risk of false positive is very high. Quickly, I'll go through the slides for the sequencing because it's a new thing. It's based on the ribosomal RNA sequencing for identifying the organisms. So we can identify slow-growing organisms, or if your sample is already exposed to the antibiotics, AFP can be identified, and fungal identification, but it's a little difficult. So here also you isolate the nucleic acid, divide them into shorter fragments, attach them to a sequencer. So all the nucleotides which are there, the sequencing is done, and you will get the results. So depending on the sequence the organism is identified, it's a very sensitive method. There are 2 types of sequencing, next generation and third generation. I won't go into the details, but only the basic principle is that the next generation is based on the fluorescent technology and third generation is based on your electrical current, where you measure the changes in the electrical current, which happen depending on the sequence of the nucleotide. So you can detect pathogen, detect the toxins, host immune response to pathogen, result within 1 hour, and drug resistant profile within 2 hours. So to summarize, summarize the bacteria, you can detect by MALDI-TOF, VITEC, PCR biofire, gene sequencing. Fungus, MALDI-TOF, the VITEC, PCR, and gene sequencing. Viruses, biofire, and gene sequencing. TB also, gene expert, XDR assay, pro-based assays, and MALDI-TOF. So in summarize, if I summarize, the newer techniques have reduced the turnaround time. These are the 3: MALDI-TOF MS, multiplex PCR, ribosomal sequencing, which help in early diagnosis and management of diseases. Highly sensitive, low specificity, false positive results can happen. Difficult to differentiate colonization, contamination, requires a huge data and analysis and genetic information. Thank you so much.